All right, gene regulation in eukaryotic cells, or eukaryotes. This works in a very similar way to the prokaryotic one, except that we have to take into consideration that in eukaryotes, the DNA is in a more complex form, right? We actually have these things called chromosomes in eukaryotic cells. So what that essentially means is that the strands of the DNA molecule are wrapped up by other protein molecules into these more complicated structures. So let's just write that down first of all. That's why this is different. Strands of DNA are wrapped up, I guess is into more complex structures. Whereas in the prokaryotes, the DNA strands themselves tend to kind of float around freely, right? They're not really, don't have this higher level of organization. And of course, we call these structures chromosomes, right? Chromosomes. And the basic structure of a chromosome is like this. You basically have proteins called histones. So what I'm going to do is draw some big blobby histones like this. Okay, then we'll put an arrow here, histones, which are protein molecules. And the DNA itself is wrapped. So here I'm going to draw the DNA molecules sort of wrapped around this, like this, or attached to it, or incorporated with it. The easiest way to envision it, I guess, is a wraparound. And it's a double strand, so now I'm going to try to draw the double strand. Ooh, that's going to be fun. So there's the double-stranded chromosome sort of wrapped around the, there, see if you can get that picture copied. Wrapped around the histones. Okay? Yes? A set length? Well, as we know from pictures in grade 11, all the chromosomes are different sizes, right? So each chromosome has a specific size, and when you look at those pictures of chromosomes in grade 11 that are already replicated, they look like that, right? And they have these bands on them. That's how they show up in a microscope. But if you dig down deeper into that structure, you'll find that what this consists of are these histones, and the DNA sort of wrapped through that. So in terms of length, I guess you could say that it's species specific, right? Every species has a particular number of chromosomes that are wrapped up. So we have some that are big, we have some that are small. The Y chromosome is actually just a little strand like that, right? That has sort of no bottom half to it. And so sizes would depend on the species and how many chromosomes there, how many DNA molecules there are. That's the way I could answer that. Okay, it's a good question. So let's get rid of that for now. Now, on the DNA, we still have to understand that along here somewhere, there is still the sections of the operon that we saw in the prokaryotes. So we still have a promoter, and we still have an operator, and we still have the genes that code for the enzyme. And upstream of that, we still have the possibility of a DNA section that codes for a repressor molecule. So we just have to envision that. To make it simpler, I'm going to change this picture. Now that you've got a picture in your mind, I'm going to just draw the histones as sort of a big red bar. Okay? And instead of drawing the double strand of DNA, I'm going to draw a single line to represent both strands. That'll make it simpler. So you can imagine this DNA sort of wrapped around. But then right up here in this section, I'm going to take a little piece of black here, and I'm going to draw a little black section. That should not go, stop right there, basically. Okay, so that would be perhaps that's the part of the gene that codes for the repressor molecule. And then the DNA would continue on, and a little further down we would see a I guess the promoter first. So then we'd see the promoter section, which I think we were drawing in green, right? And then a little further on, I don't want to draw it behind, so I'll bring it up here. We would see the operator section. What color was that? Do you remember? Yesterday in the video? Orange? Orange for the operator. So let's take this orange color. I think it was that one. And then the operator would be there. And then this wraps around. And then, of course, down here further, we were using sort of pinky colors or something for the actual gene itself, the genes that transcribe the protein. Let's use one that'll show up. Maybe that one. And there may be one or two or three, like there were sections. Oh, crap. There were sections of that. I have to press OK, I guess. So it's the same thing as yesterday, except the whole thing is kind of wrapped around. Now, because of these histones and the fact that they're all holding this all sort of together, it makes these sections of the D inaccessible. And there has to be a way to sort of get the DNA isolated so that we have to expose the operator and expose the sections. So what will happen is there will be this this molecule, or this group of molecules, and we basically call it a remodeling complex. So what happens is, in the next picture, oh, are you with me? No? All right. Well, maybe what I can do, since you're still drawing, is I will draw this complex. What color should we use? What color have we not used yet? Purple. So this complex will come along, and it's basically just a blob of proteins, okay? And they call it the remodeling complex in your textbook. Remodeling complex. Complex just means a big bunch of molecules, proteins, probably. And so what it will do is it will enter or show up at this site, and when it does, it will sort of bind with this chromosome, and it will change the shape, right? Because it's an incoming molecule, it'll push and pull and shove, and what it tends to do is it tends to dislodge the DNA from the histones. And so what happens is, you sort of can imagine that you'll have a section of DNA here, and then we'll take the histone and break it up. And so we have the DNA, and then the little piece here. But the rest of the DNA has been sort of dislodged. In particular, the promoter and the operator can be dislodged from the strand. Okay, so we'll have this complex, purple, right? So right here is where the complex would bind, and it would dislodge the histones and make I'm just going to draw the rest of the molecule here. I know, but there's, well, I guess we can do it with the pink. Why not? Pink, got to find pink. It's just a pain. And then we have the pink part. Okay. So what's happened is, and we should write this underneath perhaps, so we know what's going on. So the remodeling complex remodels the DNA or the chromosome. Sorry? Can you draw the picture? Oh, I'm sorry. Okay. The remodeling complex. All right, I'll give you a second to copy. So the remodeling complex dislodges the histones from the DNA strand. Exposing the promoter. The promoter. Promoter. With an OR? No, it's an ER. That's an E. Promoter. So now that it's exposed, once it's exposed, the repressor molecules that are present, as they were before with prokaryotes, can now access it. So you can imagine how, let's write that, once exposed, repressor molecules can access it. So I suppose what we should have written there, the promoter, let's put the promoter and the operator, because the operator, of course, is the part where the repressor molecules will want to attach. So the promoter and the operator, actually no comma necessary there. And then once it's exposed like this, we could imagine how repressor molecules could interact with it. They could interact with it in the same way as prokaryotes. They could either interact with it in a way that they disable the rest of the gene expression, or that they enable it. Remember, we talked, there's two different ways of looking at that. That's one of the things that can happen. But you can also imagine how this complex, this remodeling complex, is also regulating. It's an additional level of regulation. So the fact that you have to have the remodeling complex present, and guess what? If it's proteins, that remodeling complex could be made by another gene. So you have one gene that makes a protein that affects another gene, right? That's quite common, actually, for one gene to have an effect on the expression of another. Now, there's another couple other things that can happen, and we'll just mention them because it gets to be a little bit too complex if we get into them in too much detail. Let's see. What can also happen? Let's put two other things. Let's do it like this. Two other ways to regulate the expression of the gene. And one of them is you get this molecule, and what it does is it adds an acetyl group. So a molecule adds an acetyl group. That's a little functional group. It's basically just some C's and H's, right? And maybe an O in there. So it adds an acetyl group to the histones to loosen them. All right, I'll let you catch up to it. Okay, so these molecules are sometimes called activator molecules. They may act in conjunction with the remodeling complex or not. And often the code for these molecules is further up the same sequence, right? Further up the DNA. So it's copied in a different spot. It's kind of like the spot where the copies for the repressor. It's further up the gene, right? Further up the DNA. And that's another way to sort of get the histones to loosen themselves. And then the third thing that can happen is so that's one. The next one is what we call methylation. It's a common way of, you know what a methyl group is, right? So we can add a methyl group, and it's usually added to the cytosine, I think. I'll double check that. Yeah. So a methyl group is added to the cytosines bases in a particular location. And when it does that, of course, it changes the shape, and this inhibits. Methylation inhibits gene expression. Methyl groups are CH3 groups, right? CH3s that are being added. And basically, it just changes the shape and inhibits the ability of the molecule to be read or to be expressed. So that's another thing that can happen that we see in eukaryotic cells. We don't see it very often in prokaryotes. And the reason being is that this extra histone complex that everything is tied to, right, actually makes it harder to access the DNA. And not only harder, but it makes it more, there are more ways now to access the DNA. So that's really all we're going to say about eukaryotic regulation. These are things you'll study in more detail if you go on into genetics later. But just so you're aware of that there are some differences and that's kind of what they do. All of these you can imagine, they all work the same way. They basically change the shape of a molecule and either prevent or enable it to have the RNA polymerase attach, which can then run down and start coding the messenger RNA for those operons in the, those sequences in the operon that code for proteins. And we'll stop at that.